show a negative mean change in mass approximately -0.2 g. Direction of movement. Water moved out of the potato cells and into the salt solution by osmosis. Concentration gradient. This occurred because the salt solution was more concentrated, lower water potential, than the inside of the potato cells. Mechanism. Water moved through the partially permeable membrane of the potato cells. 5.3 Explain why the result for the potato pieces at 1.0 mol decimeter cube was different from the result at 0.6 mol decimeter cube. 2 marks. Increased mass loss. The potato pieces lost more mass at 1.0 mol decimeter cube, approximately -0.3 g than at 0.6 mol decimeter cube. Reason. The 1.0 mol decimeter cube solution is more concentrated, creating a steeper concentration gradient, so water leaves the cells more rapidly or in larger quantities. Let's move to the next slide, which all about pathogens. The question 6 is about pathogens and antibiotics. As a scientist investigates the antibiotic resistance, and you are asked to explain the results and as well as memory recall and name one antibiotic. Let's start with question number 6.1, which asks you to name one antibiotic. The answer is penicillin. Question part 6.2, why is the student wrong about disc C? Is a classical AQA command word where examiner check your analysis and interpretation of data while asking to explain the students was wrong using the figure 5. And the answer is there is a clear zone, a zone of inhibition, around disc C that shows bacteria have been killed or growth inhibited. Exam top tip. Bigger clear circle equals more effective antibiotic. Next question 6.3 asks you to suggest why concern over antibiotic resistance. Infections become harder to treat. Recovery takes longer. Mortality rates can increase. Next slide starts with viruses saying they cannot be treated with antibiotics. Now first part question 6.4 asks you the command word suggest why can't viruses grow on agar. Viruses need living host cells to replicate. Agar is just nutrient medium. Next question is part 6.5, which asks you why is it difficult to develop antiviral drugs. Viruses live inside human cells. Drugs may damage our own tissues while trying to kill the virus. Moving on to the next question, part 6.6 asks which disease caused by virus damaging white blood cells, and the answer is AIDS, and the name of the virus is human immunodeficiency virus, HIV. Let's move to the next slide where AQA examiner check your data analysis and scientific knowledge through investigation of photosynthesis factors on leaves such as starch test and limiting factors. Table 3 shows the details of each test. If you observe the statements and analyze them, followed by answer to question part 7.1, explain the results of 3 leaves. Leaf 1. No starch because black paper blocked light. Leaf 2. No starch because plastic blocked carbon dioxide. Leaf 3. Starch present because light, CO2 and water were